Thanks, Lisa, and thank you so much to C&EN News for selecting me for this award. It's a very great honor, especially hearing everyone else's exciting talks. It's so nice to see how everyone uses chemical techniques in many ways to make a big change. So I was asked to talk a little bit about my journey. When we were small, there's this cliche that people who are destined to become scientists, when they're small, they're the kind of kids who ask a lot of why questions. You know, the kind of questions, the ones that when kids say, "Mommy, why is the sky blue? Daddy, why is water hot when you put it on the stove?" There's this cliche that if you're not that kind of kid, you're not going to be a scientist. And I'm here to say that's wrong. Because actually, I don't ask why questions in my group. I ask where questions. So much of my research is focused on the idea of location. Because I think if you understand where things happen, then you can start understanding why they happen. So for example, if you understand where Leishmania parasites are they going to the skin? Are they going to the liver? Then you can start understanding why some people have mild disease versus some people have severe disease. So where is actually our route to understand disease symptoms. But then if you understand disease symptoms, you can find new ways to intervene. So where is actually leading to how to treat disease. If you understand how location relates to disease severity, you can understand how to diagnose disease. So where, again, leads to how to diagnose, how to prevent diseases. And then beyond looking at our individual location, where you are in the world is going to determine the kind of disease you get. So if you're born in the Amazon jungle, you might be more prone to infectious disease, but less prone to inflammatory conditions than if you're born in a big city. So the kind of diseases we develop, the outcome of those diseases, is strongly related to location. Now, it's a bit ironic that so much of my research focuses on location, because when I started my research journey, I actually didn't go very far geographically. So this is a map of the island of Montreal. The red dot is where I was born. The black circle is where I did my PhD. And so they're basically on top of each other on this view. And it's not just because it's zoomed out, because if you zoom into the street level, you can see it's basically down the street, less than 1 block from each other. And so I was born in one of McGill University's teaching hospitals, and I did undergrad and PhD research at McGill University. So starting first in the lab of Martin Olivier, so he gave me my very first chance when I had absolutely no lab research experience. And then I did both undergraduate honors research and then my PhD in the lab of Dr. Greta Matuszewski, already looking at the idea of location, in that particular case from a parasite point of view. So understanding how Leishmania parasites go to different body parts and how this localization relates to disease symptoms and vaccine development. I did eventually leave my small corner of the world, so I moved to San Francisco to do a postdoctoral fellowship in Jim McCarroll's group, first at UCSF, and then the whole lab moved to UC San Diego. And so there, the main interest was on using our knowledge to do drug development. So I did a lot of high-throughput screening, learning preclinical models of disease. Another big advantage of being in Jim's lab is that he gave me a lot of freedom to explore other fields. And so I wanted to highlight the environment that Dr. Peter Dorstein creates at UCSD through the Collaborative Mass Spectrometry Innovation Center, because he's created an environment where people like me can go, learn metabolomics, learn data analysis tools. And so then building on all of this experience, this idea of location, the drug development experience that I gained from Jim, the metabolomics experience that I gained from Peter, essentially what I do in my research is synergize all of this to relate location to disease development, understand disease, and then use that for drug development. And so the approach that we use is something that we call chemical cartography. The idea of making maps of molecules to find out their location. And so if we're applying chemical cartography to an organ system, we'll section that organ, build three-dimensional maps that show the position of each of these organ sections in 3D space. And then from each of those segments, we'll extract metabolites, perform liquid chromatography tandem mass spectrometry, and then computationally rebuild this data back into 3D so that we can see where those molecules are in comparison to where pathogens are located, where tissue damage is happening, where immune response occurs. And so we build lots and lots of maps, lots of 2D and 3D models. So in this case, this would be a 2D model. If you think it looks like a blueprint, that's because it is. We're mapping molecules in the workplace environment, what's found on high-touch surfaces, things that you might be touching, contacting every day. Maybe less so now, but in the past certainly been bringing up to your face, to your eyes, to your mouth, and how that could relate to occupational health issues. We're also building something that's very topical right now, 3D lung models of metabolites versus virus load. We can build models of entire organ systems, so maps of the mouse gastrointestinal tract going all the way from the esophagus at the top, the stomach, small intestine, large intestine, all the way through the anus. We've built maps of the heart. So many different organs, many different diseases are health issues. So today I'll only give you one example, focusing on what this particular model right here can tell us about Chagas disease. Now, before I can tell you what we can learn from the 3D model, I think you need to know what is Chagas disease. And so usually when I present, almost no one has heard about Chagas disease. And that's because it's considered one of the neglected tropical diseases, strongly associated with poverty, populations that are often considered invisible. So Chagas disease is caused by parasites called Trypanosoma cruzi, and T. cruzi infects over 5 million people worldwide, so numerically a very big problem. Causes mortality, causes significant health problems, but because it's so tied to poverty, it's so invisible, the current drugs we have don't work very well in Chagas disease, and so there's a need to better understand this disease and find new ways to intervene. The other reason why Chagas disease is well suited to understand the relation between location and disease is because symptoms happen in 3 very specific places in the body: the heart, the esophagus, and the large intestine. And within those body parts, damage happens at even more spatially restricted localizations. So for example, in the heart, damage happens mainly at the very bottom of the heart, what we call the apex. So location, right? Why there? Why the heart apex? What's different there compared to elsewhere? And how can we build on this knowledge to do something about Chagas disease? So that's where our 3D models come in. What we can do is we can build models of where parasites are versus where damage is happening. And what we find is actually quite striking. We find that the location of the parasite is distinct from the location of metabolic alteration. So the parasite causes damage, causes inflammation, the host tries to respond, and it significantly changes metabolism. Where does it change metabolism? In the lower parts of the heart. So that's one mechanism to explain why damage is happening at the heart apex. And it also explains why killing parasites might not be sufficient to clear this disease. So what kind of pathways are being changed? So here's an example of just one. So in the bottom, you have the structure of one particular acylcarnitine. These are conjugates of fatty acid with carnitine involved in energy production, involved in mitochondria function. And so what we find is that infection decreases the levels of acylcarnitines in specific parts of the heart. So now we've found localized effects. Now the time comes to do something about it. So if acylcarnitines are changed, what happens if we modulate carnitine metabolism? So normally in this model that we use here, all of the animals die or have to be euthanized within after infection. If we treat the animals with carnitine in drinking water, none of them die in the acute stage of disease. So by modulating this pathway, we've completely prevented mortality, but what's more striking is we've done it without changing parasite loads. So we have luminescent parasites that let us monitor how many parasites are in our animal model, where they are. So that's the picture on the right. The brighter the color, the more parasites there are. Top and bottom pictures, can't really tell them apart. There's a lot of variability, but it's the same in both groups. Localization is the same. So what we've done is induce disease tolerance, prevent mortality without changing parasite load. And since this whole story started in metabolism, we went back to metabolism again to understand its mechanism of action. So infection, acute stage infection with T. cruzi strongly perturbs metabolism. So that's why our green spots look very distinct from our orange spots. But if we look at the metabolism of our animals treated with carnitine, even though they have as many parasites as our vehicle control, metabolically, they look as though they were uninfected. So what we've done is reset the infection-induced metabolic damage caused by the parasite. And what that leads to is improvement in cardiac function, reduction in cardiac stress and cardiac strain. And so what I want you to remember from today is really the idea that making these molecular maps help us understand organ physiology. We can understand healthy organs, why they function, what is the difference between one organ versus the other. We can understand where tissue is being functionally perturbed by disease. And we find that that's not necessarily where the pathogen levels are highest. So this is our where, right? Where are things being perturbed? But that leads to why. How and how? How does disease progress? How does the microbiome interact with host metabolism? And then it gives us actionable information to develop new treatments, to understand vaccine efficacy, and to find new diagnostics. And so to build these models, we use many different collaborations. We run our T. cruzi animal models, preclinical models in our own lab, but then to apply this to other systems, I've been fortunate to have many different collaborators. In this slide, I'm actually only showing the ones from today's data, but we have many more. And we're also keen to apply this to any interesting biological question that has a spatial aspect to it. I also wanted to highlight my lab, which is a picture from last year when we were still allowed to gather without masks, but particularly 4 people who generated a lot of the data I'm showing today. So postdoctoral fellow Ekram Hossein built those 3D models of the intestines that I showed you. Graduate student Danya Dean and undergraduate Gotham did those 3D heart analyses. And then Shelley Kane is our mouse expert who's involved in a lot of our drug efficacy analyses. And with that, I will take any questions you might have.
Thank you so much, Laura-Isobel. That was great. It's so fascinating to think about. We have a question from the audience asking, do organoids or synthetic tissues help you map these metabolomics or infection courses?
That's a great question, because ideally we'd want to reduce our reliance on animal models. Not fully. So that's something we're looking into exploring a bit more, how we can do this in cell systems that might be a bit more high throughput. At least with carnitine doesn't show efficacy in vitro. So it changes metabolism, but it doesn't bring it back to normal. So certainly in that context, we needed the multi-organ interaction to make it work. But yeah, that's a good question. We'll see. I think perhaps if we work with organoids or primary cells, something that's closer to biology, but I don't think it will ever replace the complexity of a system, because if you think about it spatially, there's so many contexts, there's so many interaction with multiple cell types, but there's also spatial constraints in terms of how far the heart can stretch, how far it can contract, where things are in the body and how they relate to each other. So big questions still to explore, I think, with this.
Well, thank you so much. I am trying to resist my urge to ask a question about that virus lung slide, but we're about out of time, so I'll have to pick your brain about that at some other time. I'm sure I'm not the only one wondering about that. But thank you for a fascinating presentation, Laura-Isobel.